Hello, this is Nicolás, the DeepChain lead at InstaDeep.
Hi, I'm Marcin. I'm bioinformatics lead at InstaDeep.
We are here at InstaDeep headquarters in London to tell you more about this exciting platform. So, what is DeepChain? DeepChain is our platform for protein design. Given a complex you are working on, DeepChain will deliver insights based on state-of-the-art biophysics and machine learning models. So what does DeepChain actually do? Well, DeepChain is essentially three things. It is a scorer mechanism. So we take simulations, we take neural networks trained on millions or billions of amino acids to give a score for a given pose or sequence. But it's also a very powerful optimizer. We use methods like reinforcement learning optimization or evolutionary algorithms to find the best mutation that optimizes your structure. And finally, it's a platform where you can really leverage the power and the insights of language models trained on millions and billions of proteins to help you with the protein design task.
There are plenty of methods available online to perform bioinformatics tasks. What sets DeepChain apart? Well, DeepChain is different from what you have seen so far. DeepChain has a first principles-based macromolecular simulator which uses not only biophysics but also augments it with state-of-the-art machine learning. It gives you a possibility for independent verification of your scientific hypothesis, and it gives you infinite expansion capabilities that we'll talk about in just a moment. If I'm an experimentalist with no real computer science background, what benefit can I get from DeepChain? Well, you're in luck, because you're exactly the person that we designed DeepChain for. DeepChain allows you to tackle complex biological problems without having to know anything about optimization, simulations, machine learning. We make state-of-the-art tools available to you at the click of a button, with reasonable defaults in such a way that we know that the results that you get will be meaningful. And not only meaningful, they will be interpretable. They will inform your future experiments. And if you really are averse to computational methods, it's really a click and forget approach. And that will give you your next figure, next plot, next data set for the upcoming paper.
You can start uploading your PDB file. This can be a file available locally in your computer or any protein data bank ID available on PDB. And with that PDB file that would represent the complex, we can select some positions using DeepChain user interface and optimize them by the use of the AI designer. The AI designer will start replacing the residues in the selected positions until the binding stability or any criteria of your choice gets optimized. This is the magic of DeepChain. You are deciding your own criteria. But also, you can use the DeepChain playground. This is leveraging language models that can identify high-function regions in your protein just by looking at the protein sequence. Now I would like to show you a bit how DeepChain looks. I am in the landing page deepchain.bio, where we explain a bit of the three main modules that we described during the talk and how does it work. Once you log in, and by the way, login is for free, what you will see is for any project that you create, a 3D window where you can see essentially any structure that you download from the protein data bank or a local PDB file that you would like to upload. For this demo, we can create a new design where we are going to download from the protein data bank 6LZG, which is the RBD from the COVID complex with the ACE2 receptor here in green. On the AI designer, which is one of the modules, is basically we can select some of the positions that we would like to redesign, and we can do this through the user interface, or we can auto-select those positions for given positions close to the interface, and then run an experiment. And the experiment essentially will, we need to define out of these 25 selected positions, how many you would like to mutate. Let's say we want to redesign 4 of them maximum, and we want to launch it for 6 hours. What we want to do is improve binding, because we are maybe redesigning a therapeutic, but we could abrogate binding as well on our energy scholar. And we can use any app available on the app hub to use this. Maybe let's select 2 apps, one that gives us, for example, maximizes the log likelihood of a sequence, and something that, for example, like reduces or maximizes the probability of a benign mutation. So we run this, and what DeepChain is gonna do is it's gonna go and try to find these mutations in these selected positions that optimize for this criteria that we selected. Of course, this takes a few hours, so I can go and have I show you some results of already runs that we launched before. And what you see essentially is a scatter plot with different features. Like here we see stability versus polar contribution, but we would like to see something like binding. And here we can look for mutations coming from DeepChain that score quite well on stability and on, for example, binding. And essentially, this is what DeepChain is proposing. And here we can see some sort of analysis on this proposed mutation. And this is on the AI designer side, but we can also show you the playground. And the playground essentially, as I said, it shows a distribution of all of the amino acids probability for a given sequence. Here, for example, the spike protein sequence that we uploaded. And we see that the transformer thinks that M is highly likely to be methionine, the starter amino acid in a sequence, although here the natural one is T. And we can put all these probabilities on top of 3D structure, and this is part of the playground in the structure playground. And you see here how this looks, and we could identify zones where the probability of mutation is very low, so the wild type scores very well, but some other places where the probability of mutation is quite high, like for example here. And this is how we hope you will interact with language models in 2D or in 3D. And last but not, let me just show you something else on the playground, which is that you can input any sequence that you want. And here I have the ubiquitin sequence, and I can cut it and paste it onto the score. I can select a model. Let's take a Facebook ESM model, and we can submit this for scoring. And you can see, for example, I can also do the same for Broadbird. Yeah, and this is gonna be running on our server, scoring all the probabilities, and hopefully giving you back the results. And you see here again, M is the most likely amino acid, but here is the wild type. There is a lot more to do with this. We also see the log likelihood of the sequence. And finally, there is the MD simulations module. Again, it takes a bit to run a GROMACS, but here I have run a few simulations, and I can show you how this looks like. This is more than a 3D video. You can see here the potential energy or the kinetic energy, and you see the trajectory. But yeah, we can change, for example, the representation. And so it's a bit more than just a video. And of course, we can export this animation and do a nice plot for your research. So that's all in a nutshell of what we wanted to show about DeepChain, but hope you can try it on yourself. Just find us at deepchain.bio. Thanks.
But what if I'm a computational biologist and I develop my own methods? Is DeepChain relevant to me? Right, we're at ISMB. Pretty much everyone here develops a method or two. DeepChain has been developed for people like us. Not only it presents a simple user interface, but also it gives you an almost infinite customizability. you're able to develop your own software, your own scripts, your own methods using DeepChain, utilizing our fully free API. DeepChain is fully modular, and as such, if you want, you can replace any or every bit of the system with something that you have a trust in, something that is most suitable for your own purposes. Finally, DeepChain starts with a large ecosystem of apps that you develop and that then can be seamlessly integrated into DeepChain as a platform.
So, what are DeepChain apps? We have been talking about them. DeepChain apps is a collaborative framework that allows the user to train neural networks based on protein sequences. These neural networks can be either predictors or classifiers, and they become the scorers of DeepChain. So given a sequence, you can score things such as immunogenicity or toxicity of a sequence. So, in summary, we can take our own data, calculate the protein embeddings with the DeepChain, and use that to train a classifier or a predictor to use as an app. This app will give a score that can be used for an optimization run on DeepChain. So how can we build an app? Well, we got you covered here because we are open sourcing 3 repositories. Biotransformers, who make it very easy to work with these large language models. Biodatasets, where we are planning to collect sequences and sequence embeddings from a lot of different data sources. And DeepChain apps, where we provide the templates that make creating an app very easy. To find out more, just go to our GitHub, DeepChain Bio. They are Apache licensed, so you can use them for anything you want, not just on DeepChain. Well, we talked a lot about AI Designer, we talked a lot about language models available on DeepChain Playground, but DeepChain is more than that. There are different modules, such as the MD simulation module, where you can use GROMACS as a service. You upload your PDB structure, you set parameters such as temperature and duration of the simulation, and we will run it on our powerful NVIDIA A100 infrastructure to get the results back to you. Another important feature that we provide on DeepChain is Jupyter Notebooks hosted on DeepChain. So you can have all your data in the same place. We are looking forward for you to test it on deepchain.bio.
But what if I don't do protein design? Should I care for DeepChain at all? Well, even though DeepChain has been developed as a protein design platform, it's not only one thing that it does. What we can do is we can do protein structure assessment. You can upload a PDB file, and it will tell you what's right and what's wrong about it using our own internal assessment methodology. You can in turn do modeling of SNPs. You can see which polymorphisms have a positive and negative effect on your structure. By default, in order to provide the best possible accuracy and performance trade-off in DeepChain, we focus on the amino acid-based systems. However, within the DeepChain app ecosystem, we're able to handle a vast array of computational biology tasks. And as our simulator is very much physics-based, we're able to accommodate anything that you may want to simulate. So does that mean that DeepChain performs docking? No, not really. However, if you do know how a complex is about to assemble, you can simulate docking using restraints. And if you don't know how your complex is to assemble, you can use our language models to predict the complex interfaces.
Another important feature is the pattern search engine, where we can provide information about the IP on the sequence that DeepChain came up with. So why DeepChain? Well, we all know that protein design is a very complex problem, and it can be tackled from several angles. This is what we are trying to do on DeepChain. We are using state-of-the-art biophysics and thermodynamical models through simulation and energy potentials, but we are also leveraging cutting-edge AI, such as the transformers trained on millions of protein sequences. This conglomerate of techniques make DeepChain a very powerful tool, and we put it in your hands and accessible to the general biology community. We think it will grow with the user base and with your feedback. So, help us democratizing science and putting these very powerful tools in your hands. Join us at deepchain.bio. Remember that DeepChain is an evolving product, so we have things like docking and ab initio folding in our roadmap.
Thank you for your attention, and I hope that you enjoyed hearing about DeepChain as much as we enjoyed building it.
Yes, remember to check us out at deepchain.bio or at our GitHub open source repositories. I hope you enjoy the rest of ISMB.